Thank you very much, Brenda, and welcome back to everybody. So just to recap, yesterday we spent a lot of time covering clinical trials in some detail. We spoke about the design and analysis of clinical trials, how they support the safety and the efficacy of medical products, and we discussed their importance in special or specific populations. Today, we're going to change the emphasis a little bit, and our first session is focused on the early clinical or non-clinical studies that are done to support the use of an investigational product. These are probably studies that clinicians are less familiar with, generally reported in the investigator's brochure, which clinicians are expected to read. So I think you will find this quite informative. We're going to begin with a discussion on chemistry and manufacturing controls, which you will learn a little bit about the actual properties of the investigational product. And that lecture will be given by Paresma Patel. We will be moving on to a lecture on pharmacology and toxicology in the investigator's brochure. And that lecture will deal with some of the animal studies that support the use of medical products in a trial and some of the other non-clinical studies. And finally, we will turn to the clinical pharmacology. This lecture will be given by Shirley Xiao, and she will be able to tell us all about how the body processes the investigational product. So without further ado, let me hand it over to Paresma Patel to tell us about CMC. Thank you.
Good morning, and thank you for joining me. My name is Dr. Paresma Patel, and I'm a Division Director in CDER's Office of Pharmaceutical Quality. Today, I'll be giving an agency perspective on chemistry, manufacturing, and controls, regulatory considerations, and resources.
By the end of this presentation today, you should be able to understand the regulatory definitions and requirements for drug substances and drug products, describe chemistry, manufacturing, and controls information for IND submissions, and name some potential CMC safety concerns for INDs.
Today, I'll start off with a brief definition of pharmaceutical quality. I'll discuss how chemistry, manufacturing, and controls, or CMC, changes through clinical development. I'll provide some key regulatory definitions. I'll touch on some specific considerations for drug substances and drug products. And I'll end with important guidance documents and resources. I'd like to start with an overview of the mission and vision of CDER's Office of Pharmaceutical Quality. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. One challenge to the manufacturing aspects of an application is that it covers the entire life cycle of the product and is often rapidly changing from an IND stage of development to commercialization. During preclinical stages of development to the point of IND submission, there is typically limited CMC information. As a product's development proceeds through the phases of clinical development to pivotal clinical studies, significant CMC development is happening. CMC development has to be at a stage to assure consistent manufacturability to support a marketing application. Following approval, the CMC information continues to change as suppliers or manufacturers may change, and the manufacturing processes may continue to be optimized. Throughout this process, the CMC team is meeting with sponsors to advise on key aspects of CMC development as they move closer to commercialization. This includes early-stage interactions such as Interact and Pre-IND meetings. The CMC team is involved for Type B meetings after each phase of development, and CMC-focused meetings can happen throughout clinical development. Most CMC-focused meetings typically happen around the end of Phase 2. I'll provide a couple of key regulatory definitions that can be found in the Code of Federal Regulations. Shown here is the definition of drug substance, and to summarize, the drug substance is the active ingredient that provides the pharmacological activity or direct effect for the diagnosis, cure, mitigation, treatment, or prevention of a disease. This is essentially the active component of the drug product that affects the human body. The drug product is the finished dosage form, and it contains the drug substance, and it may or may not contain other ingredients. CMC information required for an IND submission are outlined in the Code of Federal Regulations. The main text of the CFR can be broken down into a few requirements for CMC. They include description, composition, and controls of the drug substance and drug product, information on the manufacturer, the manufacturing process, and data to support stability, information to assure identity, quality, purity, and strength of the investigational drug. The regulations specifically emphasize the identification and controls of the new drug substance and raw materials for an initial Phase 1 submission. Guidance documents are routinely published on FDA's websites. These documents provide FDA's current thinking on a topic and outline information that should be provided in IND and NDA submissions. Guidance documents clarify the type, extent, and reporting of CMC information to ensure quality of the proposed clinical studies. These documents help to outline the data needed for regulatory development and are only recommendations. If sponsors veer away from this advice, they should provide justification and scientific reasoning and meet with FDA if needed.
I would like to also take a minute to highlight that the IND assessment teams are multidisciplinary in nature. The teams include members such as clinical reviewers, clinical pharmacology, statistics, pharmacology toxicology, in addition to potential consults to other centers and other disciplines as needed. During the IND review for an original IND or first-in-human Phase 1 studies, there typically may only be 1 to 2 CMC or product quality reviewers on this team, depending on the specific product being reviewed. However, as development proceeds beyond those early stages of development, the product quality team expands to include sub-discipline expertise. So by the time a marketing application is submitted, the product quality team includes members from the following disciplines: drug substance, drug product, biopharmaceutics, which really considers in vitro dissolution of the final dosage form, drug product manufacturing, evaluation of facilities, microbiology, and finally surveillance and inspections that may be needed for the facilities that manufacture the drug substance or the drug product.
I'll start off focusing on the drug substance or active pharmaceutical ingredient information that FDA generally expects as part of an IND application. First is general information. This contains information about the structure, molecular weight, formula, and the source of the drug substance. The manufacturing process section includes a description of the chemistry for how a drug substance is made and includes both a flow diagram and a written description for each step of the manufacturing process. The characterization data section includes both structural characterization data and characterization of physical-chemical properties. The impurities section provides available information on observed and potential impurities. Sponsors should specifically address differences in impurities between toxicology and proposed clinical batches. The controls part of CMC defines a release specification, which is a predetermined set of criteria used to release batches of drug substance for use. This section includes information on the batches intended for use in the clinical study and those that were used in the toxicology study. And finally, data to support stability for a drug substance should be provided in the stability section. Preliminary stability data is typically sufficient for an initial IND submission.
One key aspect of our regulations is that they are broadly applicable to APIs with significantly variable structures. As part of regulatory submissions, information on the source of drug substance is submitted, and there are a wide variety of APIs that are regulated as drugs that we review in our office. There are drugs manufactured using chemical synthesis, which includes small molecules. Shown here are just a couple of examples: Elaxacaftor and nirmatrelvir. Additionally, peptides can also be manufactured using chemical means, such as solid-phase peptide synthesis. Shown here is just an example of plecanatide. And then oligonucleotides, such as siRNAs or antisense oligonucleotides, also fall under this category and can be manufactured using chemical synthesis. Shown here are just the two examples: patacerin sodium as well as tofersen. There are also those drugs derived from fermentation products, such as erythromycin, or semi-synthetic drugs such as paclitaxel, where the portion in red is isolated from the U tree and chemical synthesis is used to modify the structure further. There are a wide variety of drugs that are isolated from natural sources, including drugs from inorganic sources, botanical sources like the synecatechins, and animal sources such as heparin, which is derived exclusively from the intestinal mucosa of pigs. We also review combination products such as antibody-drug conjugates. These are reviewed in collaboration with biologics reviewers with a focus on the chemistry and conjugation of the drug linker or the payload linker. Hopefully the last couple of slides gave you an idea of the molecular variability and science that chemists within FDA have to understand in order to review and regulate products. In addition to the information related to the actual structure and source of the drug substance, the bulk of the review for drug substance is really the chemistry for how the drug substance is made. I'll focus on new molecular entities today because the manufacturing process is typically at an early stage of development when an IND is submitted. In order for us to assess the safety of the drug substance to be used in clinical studies, we ask for a brief description of the synthesis used to manufacture the clinical trial material. This information should be provided in both a written format as well as a synthetic scheme that provides us with a complete picture of all the chemical reagents, solvents, catalysts that are used as part of the manufacturing process, and information on any of the controls for the purity and quality of these materials. In addition, as part of the manufacturing section, we often get information on any preliminary and process controls, which are really the analytical test to ensure reaction completion as part of this at each step of a process. For a first-in-human NME IND, the emphasis really is on the difference in the quality of toxicology batches that were used for non-clinical studies and the clinical batches planned to be used. And we work closely with our pharmacology and toxicology colleagues to assess any differences between these manufacturing processes or the quality of these batches. I would also like to just emphasize that FDA does not expect that all of this information is fully understood at the time of IND submission. We're aware that the manufacturing process is being optimized for scale, yields, purity, and to minimize use of toxic or dangerous chemicals. Additionally, we're also aware that minimal steps are usually conducted under GMP at these early stages of development, and the commercial GMP manufacturing process has not been developed or finalized quite yet. FDA does encourage that sponsors initiate a CMC-focused meeting prior to initiation of pivotal clinical studies to really address any CMC challenges or to come to agreement on any specific CMC aspects related to manufacturing of an API. Next, I'm going to move on to talk about the characterization that's expected for a drug substance. I'll group this into two categories: structural characterization data and physical chemical characterization data. First is the structural characterization data. This is the data to support the proposed structure of the drug substance itself. And we recognize that structural characterization of the drug substance may be limited early in development, but as part of this characterization, we need enough data to assure identity, and we want to see submission of multiple characterization techniques to support the structure of the drug substance. Some of the standard techniques for small molecules include spectroscopic data such as NMR, UV-Vis, or IR. I would also like to note that for complex drug substances, we typically expect additional characterization. For example, for a peptide or oligonucleotide, we would expect characterization to confirm the sequence. As part of the drug substance characterization data, we expect submission of the raw spectral data itself. In addition to the raw data, we expect interpretation of this data as well. For example, assignment of peaks for an NMR or functional group assignments for an IR. During review of the IND itself, the FDA reviewers will then evaluate the totality of the characterization data and the provided interpretation, and will assess that information independently and provide any comments or developmental phase-appropriate advice moving forward. We acknowledge that identification of some peaks may be ambiguous early in development, as sponsors may be in the process of identifying impurities that are present at low levels. I want to briefly describe some of the physical-chemical characterization data that should be considered in the context of its criticality to the final drug product and drug product formulation. These types of attributes include physical attributes, including appearance and physical form of the drug substance. For example, is the drug substance a solid or an oil, and what color is it? We would want information on solubility in aqueous solutions, as well as any of the organic solvents that may be used as part of the manufacturing process. Information on particle size distribution may be limited at an initial IND submission, but the potential impact of this attribute for the drug product should be considered as additional clinical batches are manufactured. Information on polymorphic forms, as well as hygroscopicity, or the propensity of the drug substance to absorb water. Information on whether or not the drug substance is hygroscopic should be provided, and an assessment of the implications for the final drug product dosage form should also be considered. We generally encourage that sponsors monitor and characterize these attributes through development and assess the criticality of these physical and physical-chemical attributes to the final drug product, with an understanding that there may be changes to the drug product in early development. As part of the characterization section of the drug substance, there's typically an assessment of impurities. Generally, an impurity is any component of the drug substance that isn't the drug substance itself, and we typically want to minimize the presence of impurities in drug substances. Shown here is one relatively simple step of a drug substance manufacturing process to illustrate potential sources of impurities. Shown here is the reduction of a nitro group to an aniline using palladium catalyzed hydrogenation in the presence of hydrogen gas and ethanol as a solvent. From this one relatively simple step, you can see that there are multiple potential sources of impurities, including a heavy metal or elemental impurity, such as residual palladium. Residual ethanol could be a residual solvent that could carry forward to the drug substance. Any unreacted starting material would be considered a potential impurity, and in this case, the starting material has a structural alert, which is a functional group that may be of concern from a genotoxic perspective. In this case, the product of the reaction is also has a structural alert and could be a genotoxic concern. As a drug substance reviewer, we're essentially tasked with looking at the controls for the starting materials, intermediates, and final drug substance to assure that all of these potential impurities are adequately controlled and they don't pose any safety risk to patients. At early stages of development, we really want to know what control strategies are available for observed and potential impurities. And while we don't expect that all of these impurities are necessarily identified as part of the initial IND submission, we do want to understand the proposed strategy to further identify and control these impurities. And again, the emphasis in a phase one person-human study is really to address any differences in impurities and impurity profiles between the toxicology and clinical batches.
Now I'll move on to the overall controls for release of the drug substance and drug product. The specification is the predetermined set of criteria used to test and release batches for clinical use. The drug substance specification will be unique compared to the drug product specification, but the overall format and presentation is similar. The specification table provides the list of test methods and proposed limits to assure that clinical batches of drug substance and drug product meet the regulatory requirements assuring identity, strength, quality, and purity. In the initial IND, the analytical test method can be briefly described or referenced to a compendial test. The limits for an IND are typically flexible based on the available toxicology and clinical batch data and are often further defined and narrowed as development proceeds and nears commercialization. Typically, we would recommend that any critical attributes for the product be monitored as a test within the specification. For example, if the particle size of the drug substance is important to the drug product manufacturing, we would recommend this attribute be monitored through clinical development. As part of the IND submission, we expect to see either tabular batch data or certificates of analysis for the proposed clinical batches, tested according to the proposed specification. Shown here is a representative drug substance specification. Specifications include three main elements: the test attribute, the analytical method, and acceptance criteria. We recommend that you capture your rationale in a thorough justification of specification document. FDA generally is more flexible with acceptance criteria for early stages of development, with the understanding that there's very limited batch data available. What we're looking for is that there is sufficient control of the identity, purity, impurities, and quality of the drug substance to assure safety. A better understanding of the drug substance manufacturing and quality is expected as development proceeds, and we would expect phase-appropriate updates to the specification table as additional process and product understanding is gathered and the development program moves closer to commercialization.
I would like to briefly discuss stability data for drug substances. Stability studies are conducted to measure how the quality of a drug varies with time under the influence of environmental factors. And these studies are important to establish a useful retest period or shelf life for a drug substance when stored under long-term storage conditions. Stability specifications may be different from release and should include attributes that are likely to change during storage. For example, attributes such as appearance, purity, water content, or polymorphism are typically considered stability indicating attributes. Long-term storage testing is conducted under the recommended storage conditions for the shelf life or retest period of the drug substance and is used for labeling. Accelerated testing is typically run under conditions with increased rates of degradation. The stability section of an IND should clarify the tests that are run to monitor stability. Preliminary data on representative material for both drug substance and drug product is typically acceptable for an initial IND submission. If stability information on the clinical batch data is available, that information should also be submitted in the IND. If stability data is not available, we ask that sponsors provide a commitment to continue testing the clinical trial material through the duration of the proposed clinical study.
I would like to now touch on specific information that is needed for a drug product compared to a drug substance as part of an IND submission. For the drug product, it's important that we have a description of the dosage form and any information to justify the safety of a novel technology or any complex drug product formulation. It's particularly important to clarify any patient administration information for the proposed dosage form. For example, a drug product that is administered parenterally, it'd be important to consider compatibility issues with any administration devices or solutions that may be used to dilute the drug product prior to administration. The quantitative composition of the drug product should be provided, and this includes a list of the inactive ingredients or excipients. We ask for information on the compendial status or quality of those excipients. If novel excipients are used, additional CMC information may be needed to assure quality of those excipients. Additional information may also be needed to assure safety of any animal-derived excipients. If using novel excipients, we generally recommend discussing your approach during a pre-IND meeting to assure adequate data is provided in the IND submission. The drug product manufacturing process should be described with both a brief written description and a flow diagram. And additional information on the sterilization process should be provided if it's applicable to your drug product. Similar to the drug substance, the control of drug product is essentially a specification that includes a set of criteria to demonstrate quality of the drug product prior to releasing batches. This section includes information on impurities that are specific to the drug product, as well as the proposed batch data for use in the intended clinical study. Finally, information on how the drug product is packaged and preliminary stability data should be provided in the IND submission.
There are a few miscellaneous requirements that are outlined in the IND regulations. One is the requirement for a product label that includes a caution statement, which is shown here. Additionally, the submission of an investigator's brochure that has a brief description of the drug substance and the drug product formulation should be provided. Additionally, the requirement for submission of an environmental assessment or a claim for categorical exclusion are also referenced here. I want to point out that INDs generally are categorically excluded from the requirement to submit the environmental analysis, but we do need that claim and request for that claim in the IND submission. Similar to the drug substance specification, the drug product specification is the predetermined set of criteria to ensure identity, strength, quality, purity, potency, and bioavailability of the drug product. The specification includes the attribute being tested, the analytical procedure, and acceptance criteria. Shown here is a representative drug product specification that includes some key tests, including identity and appearance, assay and impurities, content uniformity, and water content. In this case, we have controls for reconstitution, particulate matter, osmolality, sterility, and endotoxins, which are dosage form specific. I would like to briefly touch on the information that should be provided on the container closure for a drug product and information to demonstrate stability. For an IND, a brief description of the container closure and packaging components can be provided with any precautions that may be needed to ensure protection of the investigational drug during clinical studies. Similar to the drug substance, the drug product stability data is required to establish the shelf life and set an expiration dating period for the final product. Drug product attributes that are likely to change and impact the quality or performance of the drug product should be investigated on stability testing. Stability data should be conducted under long-term storage conditions and accelerated conditions. Preliminary data on representative material for drug product is acceptable for an original or initial IND submission. If stability information on the clinical batch is available, that information should also be provided in the IND. Additionally, in-use compatibility data should be provided to demonstrate compatibility of the drug product during administration. These studies should include any reconstitution steps and data to support hold times.
Some of the CMC issues that are likely to lead to clinical holds of ongoing studies include the following. Questions related to adulteration or purity of the materials used to manufacture the drug substance or the drug product. If the impurity profiles are not sufficiently characterized or the safety of the impurities is not justified by supportive non-clinical studies. Any impurities that are known to be highly toxic or pose potential risks to patients. Unreliable analytical methods that may undermine the confidence in the data provided in the IND. Insufficient batch data to demonstrate manufacturability of either the drug substance or drug product. Any significant stability issues. Lack of sterility assurance or endotoxin controls could pose significant safety issues or risks to patients. Challenges with the drug product formulation, such as formation of particulate matter upon storage of a parenteral drug. And finally, issues related to CGMPs at the manufacturing facilities. One way to avoid issues at filing of an IND includes consistent communication with the FDA through industry meetings and paying close attention to the advice given during those meetings. Prior to an IND submission, every sponsor is granted at least one pre-IND meeting. As part of this meeting, the sponsor of the IND submits a brief package that describes the background of the product and proposed clinical development. CMC-specific focus areas during a pre-IND meeting include the following: whether the manufacturing process and characterization data appear reasonable, the acceptability of the drug substance and drug product controls or specifications, the overall stability data and stability design, whether the impurity levels and controls appear reasonable for phase 1 studies, adequacy of the toxicology batches to support use of the proposed clinical batches, and any overall potential gaps or hold issues that the CMC team may find. I would like to end with a few resources, including important websites as resources, IND-focused guidance documents with CMC advice, and resources for expedited programs. This slide provides resources that are specific to control strategies for impurities that could be used during development.
And finally, I have two challenge questions for today's presentation. Challenge question number 1. The regulatory term for an active ingredient intended to furnish the pharmacological activity or other direct effect in the diagnosis, cure, mitigation, treatment, or prevention of disease, or to affect the structure or any function of the human body is A, drug product, B, impurity, C, drug substance, D, intermediate. The correct answer is C, drug substance. Challenge question number 2. The following are CMC safety concerns, except for which one? A, impurities of known or potentially high toxicity. B, insufficient batch data. C, lack of sterility assurance or endotoxin control. Or D, the manufacturing process route for the marketing application has not been finalized. The answer is D. All of the other options are potential CMC safety concerns. However, the manufacturing process for marketing is typically not finalized until closer to conducting the pivotal clinical studies.
That concludes my presentation, and I will be back later to respond to some of your questions. But now I will turn it over to Nicolette Beal to share her insights on pharmacology, toxicology in the investigator's brochure.